Welcome student. In the next lecture, we have started our discussion with xerophytic plants which are adapted to grow under low water availability. We have discussed some examples like medium leaves we have discussed, then casuarina stem we have discussed that how they have been modified and how some additional structures are present in those plants to help them in survive in that extreme environment. Now we will take some more plants. We will try to discuss some plants which are adapted or which are modified into certain structures which we call as phylloclade. Now, Muhlenbeckia is an example in which the stem has been modified into phylloclade. Now, phylloclade also we have discussed that basically they are flat green stem which are modified to perform photosynthesis. Now, this is a picture of Muhlenbeckia. Okay. You can see all the structures are appearing green in color, but this is the stem. Okay, you can easily make out by the presence of nodes and internodes. Okay, and you can see here from nodes leaves are actually coming out, but the stem is modified as flat structure and it is green in color, so definitely it will perform photosynthesis. Now, when we will cut the transverse section of this plant, we will see that definitely it will have thick cuticle. Okay, you will find stomata which will be distributed on all sides. Uh, Now, since this is a flat stem, so its thickness will not be very much. Okay. If you will cut the transverse section, if you are cutting it like this, okay, you will see a boundary, a flat boundary. Okay. Then after that, you will have, suppose I am okay, and then you will see a layer of cells which will be basically made up of mechanical tissues, that is sclerenchyma and collenchyma, and they will be present in patches. And you will see lot of vascular bundles uh, which will be present inside this central cylinder. Okay. So, in that vascular bundle, definitely xylem will be on the lower side and phloem will be on the outer side. Now, in this you can see that here a thick cuticle will be there definitely. Okay. Lot of stomata will also be there. Okay. Stomatal pores will be there. And then you will have a thick layer of cells below epidermis which will be arranged in patches. This will be sclerenchyma patch and this will be chlorenchyma. Okay. So, lot of chlorenchyma, definitely chlorenchyma will be there to perform photosynthesis, but it will be followed by sclerenchymatous patch. Then this central cylinder will be also surrounded by sclerenchyma. So you can see that the central cylinder in which all the vascular bundles are embedded, that will also be surrounded by sclerenchyma. Okay, and in that you will have a lot of vascular bundles. In the center you will have pith region. Now these vascular bundles, slightly the side one will be bigger in size, and this will be, the vascular bundles will be, you can say that it will be conjoined, it will be conjoined, collateral and open. That means each vascular bundle will have a phloem and it will have a cambium. And xylem. Okay, they will both be in the same radius, xylem and phloem, that is the conjoined. Phloem will be on the outer side and xylem will be on the inner side, collateral, and open means cambium will also be present. Now each vascular bundle will also be covered by these vascular bundles. You can see that they are just below the sclerenchymatous patch, which is actually covering or making a cap like structure on the these vascular tissues. Now if you draw the cellular diagram, you can easily make draw it. It's you can draw it a cuticle here, then you will have a stomata here. Okay, then you will have a chlorenchyma and then sclerenchyma. Sorry, chlorenchyma you will have, and then thick walled sclerenchyma. And you will see that these stomata will be definitely present above chlorenchyma, because sclerenchyma will not help out in any sort of gases exchange. Okay, and below this also this entire ring of sclerenchyma will be there. Okay, and below this sclerenchyma you will see vascular bundles. Okay, on that you will have cambium, then you will have xylem, and then on top you will have phloem. Okay, you can see here that below this sclerenchymatous patch, vascular bundles are present. Now this is a cellular diagram. Okay, and these are the pictures which has been taken from the microscopic slides, and you can see here that how you can see red color tissues here. These red color tissues are definitely, they are the stained lignified tissues, and definitely they will be either sclerenchyma or in xylem also lignified tissues are present, so that will appear colored. So this is the sclerenchyma, you can easily see here. Then this is the, it is followed by chlorenchyma. Okay, and then you can see here these are the vascular bundles. The small vascular bundles are embedded around this central cylinder. See, in this picture also you can easily make out the vascular bundles. See, this is all the green color chlorenchyma and the pink color sclerenchyma in alternate patches, and here also you can see that just below sclerenchyma vascular bundles are embedded, in which you can see the color of the xylem, although phloem will also be present on the upper side, and in center you can see parenchymatous pith. In this figure, this is also a figure cellular diagram. See, you can see that on the sides, okay, on the sides a very big patch of sclerenchyma is there, so that also is completely evident in the microscopic pictures, and you can see lot of vascular bundles, cambium you can see, then xylem is there, and then phloem is there. Okay, so this is the arrangement of the phylloclade of Muehlenbeckia. Now coming to the next structure, we will try to understand cladode of asparagus. Now asparagus also you will see that it will have a main stem. Okay, it will have a main stem, and all those branches will come out, although they appear like leaf-like structure, but they are modified branches. Okay, and they are green structures, they are flat structures, and they will perform photosynthesis. Now in this asparagus, two types of branches are there. Main shoot, the main shoot will have monocot like structure. See, when you will cut the section of this main shoot, in monocot you will observe that it will have epidermis, then it will have hypodermis, and then lot of vascular bundles will be embedded in this. Okay, they will be scattered, so it will have a monocot like anatomy, whereas the small green branches, okay, they will have a somewhat different structure as which is actually not present in in the stem, main stem. Okay, it will have a different type of structural organization. So when you will cut the section, you will see that it will have a boundary. Okay, definitely the boundary will have epidermis, and then it will have cuticle. And you will find stomata also, and that stomata will open into a substomatal cavity. Okay, so a cavity will be also there where that air exchange or air space will be present. Okay, so in that cavity, this stomata pore will open. Suppose if I draw two stomatas here, then followed by epidermis, and definitely it will have a cuticle. So you can have cuticle, then you will have epidermis, okay, and below that you will have a single layer of palisade like tissue. Okay, and these cells will have chloroplast and they will perform photosynthesis. So all around these palisade layer, a single palisade layer will be found. So chloroplast will be there. You can see that this is the palisade tissue. Now below this palisade tissue, you will see presence of a spongy parenchyma. Okay, they will be loosely arranged. These cells will be loosely arranged and they will fill the entire tissue. Okay, so now, and between them you will see a single vascular bundle. This vascular bundle will be surrounded by a sclerenchymatous tissue. Okay, okay, so all these structures will be there. Then in between you will have definitely xylem, and above that you will have phloem. So this will be xylem, and then you will have phloem, and then this will be the sclerenchyma. So this type of anatomical structure you will find in asparagus. Now coming to aloe vera. Now you would have seen the leaves of this aloe vera. They are you can see they are fleshy, they are thick. This internal structure contain, when you will see the internal structure, you will see that lot of water storing tissues are present. So definitely you can see that they are thick and shiny. So they will have a very thick cuticle layer. Okay, when you will cut the section of this leaf, you will see that they will have a very thick cuticle. Okay, and definitely they will have outer epidermis and they will have inner epidermis. Stomata will be located on the lower surface. Okay, then you will have some palisade like layer which will be found on both the surface. Okay, they will be found on both the surface. Okay, palisade like tissues will be found, and after palisade you don't have that spongy parenchyma. This is all palisade layer. Between them there will be lot of water storing tissues will be there, thin walled water storing tissues will be there, and in between you will have vascular bundles also. Okay, so water storing tissues will be present in this region. Okay, most of the space will be occupied by those tissues. Then coming to very important epiphytic plant. Okay, we have talked about xerophyte, we have talked about hydrophytes. Now let's try to also understand how epiphytes, means those plants which grow on other plants, how they are also adapted. Now one very important epiphytic plant is vanda, which is actually orchid. It's an orchid which grow on other plant. Now this is the structure in which you can see that vanda is growing in a pot and it's hanging, and you can see that lot of aerial roots are coming out. See, if they are growing on the plant, they are definitely not in contact with soil. So if they will come, roots will come out, they will also be hanging, and the primary function of the roots will definitely be absorbing moisture which is present in the atmosphere. Okay, so their function will remain same, but they will be adapted in that way that they can absorb moisture. Okay, see, they are not in touch with the water column. They have to absorb moisture from the atmosphere. So in that way they will be adapted. So let's try to understand when we will cut the section of this aerial root. Okay, root we are talking about, because the root will show that type of modification. We will see that velamen tissues are present. Okay, so you can see lot of elongated cells, multi-layered cells or multi-seriate cells will be there, which is known as velamen tissue. Okay, and these cells are elongated and these cells are dead. Okay, these are not living cells. Below this cells you will find exodermis. You can say that this is the outer layer of cortex, as we have discussed before also, that some cells will develop into exodermis. It's an protective layer only, which helps in not only in preventing water loss, the flow of water to the atmosphere, but it also acts as a protective covering. It is protecting the cells which are on the inner side, means, or the vascular tissue which are present on the interior. So it is also protecting these tissue. Even velamen tissue is also doing so. It is preventing water loss and it is also protecting those tissue. These velamen tissues are definitely porous and fibrous, so they will behave like sponge. They will take moisture, they will absorb moisture from the atmosphere like a sponge, and they will also protect the vascular tissue. Similarly, these exodermis. Now you can see that in between exodermis, some cells will be thin walled. See, exodermis is thick wall cells and it is suberized. Okay, suberin is there, suberized. Some cells will be thin walled, they are known as passage cells. Like in endodermis also we had discussed that it is suberized, but some cells are thin walled, and which will help in flow of water, because everything could not be blocked. And now the bulk of the cortex will be parenchymatous. Okay, lot of parenchyma cells will be there. Okay, definitely in the cortical region. Okay, then coming to the vascular tissue, see, when you will see that you will have endodermis and you will have pericycle, normally as we have in root. Okay, so its arrangement will be that of typical root, but presence of velamen tissue, that is actually, you can say, it's an epidermal layer only. It is an extension of epidermis. It is modified epidermis, you can say. So this tissue, then you will have endodermis. So you will have normal endodermis, then you will have pericycle. Pericycle also, let's draw. Then inside, in root, you have radial vascular bundle. Okay, a lot of xylem and phloem are there, which are in different patches. You will have xylem patch. Okay. Sorry, they will be in exarch orientation. Okay, so that is a difference. So we have to make exarch orientation in case of root. So that means protoxylem on the outer side and metaxylem on the inner side. So between these xylem you will see phloem also, patches will be there. Okay, so this will be protoxylem and this will be metaxylem. Then this will be phloem. Okay, and in the center you will have pith. And some sclerenchymatous tissue can also fill this space inside this central cylinder. So presence of velamen tissue is an important feature that is found in this epiphytic plant. This is the cellular diagram of Vanda. Okay, we can see here. Now this is the microscopic picture of the slide which I have prepared. So here also you can see the cortex. See, these are the upper layers. Okay, so this is the velamen tissue. And here exodermis is very clear. You can see somewhat bigger cells. Exodermis will be somewhat bigger in size. Okay, they will be thick walled. So you can clearly see the presence of exodermis. Okay, in this also these bigger cells are exodermis. And these are the somewhat elongated cells. This is the velamen tissue and this is the cortex. Okay, which is basically parenchymatous in nature. And this is the entire structure where you can see the velamen tissue. A thick velamen layer is present outside. Then exodermis is there. This is the cortex, and in the center you can see the central cylinder. Okay, endodermis is also very clear, and you can see how the arrangement of vascular tissue is there. A lot of vascular bundles are arranged in form of radial arrangement and in exarch pattern. Now coming to another important plant, which is parasitic plants. We will try to understand how the parasitic plant might be modified to perform its function. Now Cuscuta is a very important parasitic plant, and it is also known as dodder. Now it is highly parasite. Okay, it is highly parasite means it is completely dependent on the host plant, and it is so much that it does not produce its own leaves also. No leaves, no roots. Okay, so it's a leafless plant, no root, no leaves. So you can imagine how parasite it is, how parasitic it is, that it does not require even leaf. It is completely dependent on the host plant. You can see this Cuscuta. Now only these thread-like structures which you can see, that is only Cuscuta, and the green color leaves are definitely of the host plant. Now this Cuscuta is producing flowers because it has to reproduce, and it's a angiospermic plant. It's a dicot plant. Okay, it produces flowers. You can see white color flowers it is producing. So it reproduces normally, but yeah, it doesn't produces any leaf-like structure or even root. Now, how it actually performs its parasitic activities? By producing haustoria. Now, haustoria are the penetration of vascular tissue from the cuscuta inside the host plant. You can see here that how this cuscuta, this stem is winding around this host plant, okay. It is encircling around the host plant, it is producing flowers. Now you can see here that it is producing haustoria which is penetrating inside the host tissue. Now suppose this is the cuscuta plant, okay, and it is just attaching itself with the host plant. So in the host plant also you will have vascular tissue, you will have pith, and you will have xylem and phloem. So definitely this haustoria should enter, at least it should enter till phloem to obtain nourishment. So it is observed that the vascular tissue of this will enter into the phloem region, okay, they will enter into the phloem region. And they will obtain nourishment. See, this is the cuscuta stem, you can say, stem, this is the host stem, okay, this is the host xylem, host phloem, okay. So these in this way it will penetrate and obtain nourishment. Now this is the picture, this is the microscopic picture slide I have prepared. So you can see here that this is the cuscuta which is putting out haustoria up to the vascular region. You can see here that it is passing through the phloem and it is even entering to the pith region also, it is covering xylem portion. See, this is also a microscopic slide preparation. So here also you can see that the tissues are entering till the pith region. So at least they have to enter the vascular region to obtain nourishment. Now in the last we will try to understand how some special type of root anatomy, that is pneumatophore. See, in marshy places, okay, shallow places, marshy places where most of the plant roots are completely immersed in water, okay, they are placed in shallow waters. So in that shallow water sometimes high salt concentration is also there. So our plant is they are known as rhizophora. So you can see that they are growing in marshy areas. Now since their root is completely submerged in water, so these roots also require oxygen for respiration, they also require gas exchange, but since water is completely present here, so it's not very easy for them to respire, to obtain the gases. So what they do, they give out aerial roots. You can say that these roots come out from the soil to the above ground surface. Normally the roots are found on the below ground surface, but these roots come out above ground and they are known as pneumatophore. Pneumatophore, and they have lenticels, lot of lenticels, that is pores, air breathing pores are present in these roots, and with the help of them they respire. Now normally they have normal structures, okay. They are these roots are negatively geotropic and its anatomy is like that of stem of dicot. So in stem of dicot you have vascular bundles arranged in the form of ring. So they will have a structural anatomy as that of stem, but they will come out from the root. So they are basically considered as to be modified root only. You can see that when periderm will develop, means the outer lateral meristem will become active, they will form cork on the above surface, and in that lot of lenticels. So more number of lenticels will come out and they will act as a pores from where they can breathe or gases exchange can take place. Below this cork, you will have cortex, which will also contain air chambers. They will contain air parenchyma for more better gaseous diffusion and exchange. Then after that, they will have normal organization. They will have endodermis, then pericycle, and then secondary xylem and phloem formation will be there. It might be there, or otherwise they can have normal vascular tissue. So this is all about the anatomical features which a plant in which is present in the plants growing in different habitats, and how these structures are present and how these structures are helping those plants to survive in that environment. So with this we end about plant adaptation, and one lecture is remaining. In that we will discuss some more anatomical features which are present in plant that we have not discussed before. Okay, so thank you for today.